So the next type of recombination I'm going to discuss is VDJ recombination, and this occurs in adaptive immune systems when lymphocytes make proteins called antibodies or immunoglobulins to fight off antigens, which is foreign material. The reason the immune system is able to make antibodies specific for 1 billion different antigens despite only having about 3 billion base pairs is VDJ recombination, which is a somatic cell DNA rearrangement. So there are 5 main classes of immunoglobulins, which I have listed here, and they are M, G, D, A, and E. But they all have a similar structural organization composed of 2 heavy chains and 2 light chains. So here we see in black the heavy chains and in green the light chains. So each chain has a constant and a variable domain. The variable domain binds the antigen and is located in the amino-terminal region up here. So these are the 2 variable domains of each chain. And the constant domain is located at the carboxy terminal. So here's the constant for the light chains, here's the constant for the heavy chains. So there are 5 types of heavy chains, each specific to an immunoglobulin class. So for M, we have mu, for G, we have gamma, D, we have sigma, A, alpha, and E, epsilon. So and there's also 2 types of light chains, which are the kappa and the lambda chains. And they can bind to, the 2 types of light chains can bind to any of the heavy chain types, so we can see how already there's a lot of potential for diversity. First I'm going to discuss how the variable regions of the heavy and light chains are formed. Okay, so for the heavy chain, the primary structure of the constant domain defines the 5 types unique to each of the 5 classes. So in this little drawing here, I didn't include every single type because there's many as we will go over, but this is the constant domain. So here we see C mu. If this was paired with something that would make the immunoglobulin M, this is gamma, so etc. You only use one of the constant regions. And the variable domain is divided into 3 distinct subdomains. We have the amino-terminal variable region V, and there are about 100 of these. The central diversity region D, the carboxy joining region J, and each heavy chain variable region is encoded by different gene segments within the immunoglobulin gene on chromosome 14, which is what is written here. So there are about 100 V regions, as I said, 20 D regions, 6 J regions, and 9 C regions in the immunoglobulin heavy chain locus. And recombination of the V, D, and J regions provides for the diversity of the heavy chain variable region. So there are 2 proteins, RAG1 and RAG2, that stimulate the immunoglobulin gene recombination, and there's enzymes collectively termed recombinase that carry out the recombination events. And in these events, 1D region is first joined with 1J region, so 1D and 1J are joined, and then 1V is joined with the 2 of these, with a new DJ region. So there's still a small amount of nucleotides between each of the 3 regions. The light chain has a very similar process, but they only have 2 regions that make up the variable domain. There are about 100 V also, and there are 5 J regions for the kappa light chains and 6 J regions for the lambda light chains. They do not have a D region as part of their variable part. So the VJ recombination for the light chains occurs similarly to the VDJ recombination on the heavy chain. Okay, so these recombination events actually are very important because they bring the promoter, the immunoglobulin promoter, which is upstream of the V region, and they bring it closer to the transcriptional enhancer elements, which are downstream of J. So without the recombination, they wouldn't be close enough and transcription wouldn't occur. For the heavy chain, further recombination events between the switch region of C mu, so here I have drawn the switch region, the switch region of C mu and the switch region of another C domain. And like I said before, the C domain that is part of the heavy chain determines which class of immunoglobulin it will be. So mu is pretty much the first one, and then if it's switched with another one, then that one will be transcribed instead. So let's say these 2 switch regions were recombined, then the C gamma region would be transcribed instead of the C mu, and it would be an IgG, not an IgM. So next, post-transcriptional processing occurs, and this occurs for both the heavy and the light chain. And several elements are spliced out of the mRNA, and the variable region is spliced together with the constant region. Another important recombination, well, not recombination, but another important event that contributes to diversity is somatic mutation, and this occurs when the variable region can be altered to result in higher affinities for the antigen. So in summary, there are probably at least 4.8 times 10 to the 10 possible immunoglobulin combinations, so we can see how our immune system is able to adapt to almost any antigen.